off of the Eastern Viticulture Facebook page a few weeks ago. Someone asked, grapevine trunk diseases seem to be all the rage currently. What's the deal? Is this all from Dr. Smart visiting a while back? What diseases are out there and which ones are worth worrying about? So hopefully we're going to be answering some of these questions in the presentation today. The webinar that was put on in December 2018 gives a lot of detail about the biology and mechanisms behind trunk disease, so I'm not going to talk as much about that today, but just so we're all on the same page, grapevine trunk disease is a category of diseases that infects woody tissues. And they survive from year to year, and they gradually spread through the vine. So they're gradually infecting the vines and damaging the plant as they go through and spread. There are several different genera of pathogenic fungi that cause grapevine trunk disease. So when we say GTD, we're not just talking about Botryopsera, we're not just talking about Eutypa. It could be any number of genera that are causing these similar symptoms. Basically, how this works is the fungal spores enter the wood either through pruning injury, cracks, rubbing up against a wire, any sort of injury that we cause to the vine. And then they spread through the vine gradually, and the eventual result is unproductive vines. So this is a photo of a cordon, and if you look at it from afar, maybe you're just walking through the vineyard, it looks like there's a canopy there. But if you look more closely at the vine, you can see that there's not actually any fruit coming out of this entire cordon. So we've got a lot of blind wood here, no fruit, and no actual shoots coming out of this entire area. But where this gets complicated in cold climates is a dead cordon, that could be due to trunk disease. It could also be due to winter injury, or it could be due to a combination of factors. So it's important in our area not to rush to conclusions when we see a vine that is dying back and is becoming less productive. These diseases spread through the vines very slowly, and I think that's important to understand when we're trying to figure out how much these diseases are contributing to dieback versus winter injury. Because winter injury, like what we had in Minnesota last year when it got down to -38 in a lot of parts of the state, that caused entire vineyards to die back to the ground all at once. That's not something that we see if it's just a trunk disease problem. Trunk diseases can take many years to travel from the pruning wounds where they're introduced down to the trunk. And so because of this, removing and replacing failing cordons is a consistent strategy to remove disease from those plants. So I'm going to talk about the trunk disease project that we have going on in Minnesota. We're really excited to be working on this project. It started out when we did have Richard Smart visit a year and a half ago. That was summer of 2018. And as a result of his visit, a lot of the grape growers in Minnesota were very concerned about grapevine trunk disease. There were all of a sudden a lot of unknowns, and there was a lot of confusion around this problem and how much of an impact it's actually going to have. We had one grower cancel a very large vine order because they were under the impression that all of the vines were going to be infected. There were just a lot of misconceptions going around. So we needed to find out what was actually going on with grapevine trunk disease in our state at a more technical level. You do have to do lab analysis to positively diagnose grapevine trunk disease in vines. So we took one initial sample in July 2018 with the hopes that we could use that data to as preliminary data to apply for funding to look at this issue. So from that vineyard that we sampled in July 2018, we did find 3 different trunk disease genera, that should say genera there, because we found Ceridium, we found Cytospora, and we found Diatripella. We did not find any Botryopsera in that vineyard. But grapevine trunk disease are present in every grape growing region. As we know, if we listen to the December 2018 Northern Grapes Project webinar, where this that screenshot on the right is from, they're all over the place. So why do we care in Minnesota? Well, there are several reasons why we care so much about studying this. The first is we need to know what species we have throughout the state, and we need to know how those species are distributed throughout the state. We need to know, is grapevine trunk disease something that's present in all vineyards, or is it maybe worse in some parts of the state than others? Grapevine trunk diseases may, in several ways, impact vines in cold climates differently than they do in warmer climates. So we needed to look at what impact they're actually having on our vines. And a lot of the management recommendations that we hear for Mediterranean climates must be adapted to the climate that we have in colder areas, as well as the grape growing culture of our areas too. For instance, one of the recommendations we hear from some growers in California or similar regions is to paint or spray every pruning wound, which is, as you can imagine, a very tough sell as an extension educator talking to growers who have never had to think about that before. They don't want to go out and paint every pruning wound. Also, if we're pruning right now in February, and I said it was 21 degrees today, we can't go out and spray right now. So some of these recommendations really need to be adapted, and we need this data in order to do that. So we did get grant funding from the Minnesota Agricultural Experiment Station. About a year ago, we received that funding. Our project is called Determining Distribution, Impact, and Management Options for Grapevine Trunk Disease in Minnesota Vineyards. Dr. Matt Clark on the left is the PI of this project. That's a picture of me in the middle. Dr. Bob Blanchett in the plant pathology department. He has a lot of knowledge about woody plant diseases, so he's great to have on the project. And we were able to hire a grad student named Davey Ducray to work on the project as well. I want to give a quick shout out to Davey because he's the person who's doing a lot of the hard work on this project. He's the one who goes out with me to the vineyards to collect the samples, and then he analyzes all the samples in the lab, which is a very labor-intensive process. There are 3 goals in this project. The first one, which is what we worked on this past year, is to identify what species are present and where they are throughout the state. We have not just collected samples from Minnesota, but some from Wisconsin and Iowa as well. The second goal is to learn how much impact grapevine trunk diseases have on cold climate vines. You know, there's this question of, is it winter injury or is it trunk disease or is it a combination of both? That's the question that number 2 is trying to get at. Number 3, from the data and the experiments that we do in the project, we need to be able to develop grapevine trunk disease management recommendations that are relevant and feasible for cold climate vineyards. So the first step in this process is sampling wood and getting samples from the vineyard. The second process is fungal isolation. So once we get those samples from the vineyard, Davey has to grow those in the lab to isolate the fungus from the samples. And then he has to identify what it is. From there, we look at the fungal species, see if it's a pathogen in the first place. Some of them are not pathogens. And then research more about that disease and see how it's actually impacting the vines. When we go out to vineyards, a lot of the time, I'm the one talking to the growers and answering whatever questions they have, not just about trunk disease, but anything. And Davey concentrates on collecting those samples. So we try to sample from vines that do appear to have symptoms of dieback, but we also try to sample from vines that are very healthy. So that photo on the left that we took a couple of weeks ago, it's a very healthy vine. And the reason why we need to sample from both healthy and unhealthy vines is to answer that question of how much impact is trunk disease actually having. If we find that there's a lot of trunk disease in very healthy vines, then that may be evidence that, well, it's there, but the vines are still healthy despite it. It doesn't necessarily mean that those vines are going to die or be unproductive. So when we take a sample, we take a photo first, and that way we can correlate the sample to the state of the vine in the vineyard, whether it's healthy or not, or how old it is, things like that. Look at the inside, and this is a symptom that we are looking for. Grapevine trunk diseases can take on a lot of different appearances within the vine, but usually it's some sort of discoloration. There have been a few times where we don't actually see trunk disease symptoms on the vine, but we do find it when we analyze them in the lab. Here's a few of the most interesting cankers that we have found. We see these a lot, but so sometimes it can be a rounded pattern, sometimes it's in a V shape. The one on the right, it actually infected half of the cane and not the other half, split at about 50-50. So if you look at the photo on the right, you can kind of see how the mechanism by which these are acting. They're cutting off vascular flow to the rest of the vine. Here's a snapshot of what we've done so far in the project. We have gotten 170 vine samples from 35 different vineyards throughout Minnesota, Wisconsin, and Iowa. And from those, Davey has isolated 176 diseases and sequence those. 79 of them have been non-pathogenic fungi, and 42 of those have been unknown species. And as far as we know, this is the largest grapevine trunk disease survey in the US. We're pretty sure, just because of the volume of vines that we have sampled in the project so far. This is a map showing where we have sampled and where we still would like to sample if possible. All of the counties in yellow are where we have confirmed pathogens. The green county means that we did sample there, but we've only confirmed a non-pathogen. That was very rare. Almost all of the vineyards we sampled did have a pathogen confirmed. And the dark purple vineyards are places where we have sampled, but we're still working on the results. So once we get that sample into the lab, it's Davey's job to analyze it and figure out what species are in there. So he cuts the canes into very, very thin cross sections and then basically puts those onto plates and identifies what they are. So here's those cross sections. Then he takes a cross section and cuts that up into very, very small pieces like that. Puts those pieces on an agar plate. And what this does is it grows out whatever fungus or whatever other microbe is in there. Here's about 3 weeks after he plated those pieces, and you can see several different fungi growing from those little samples. So sometimes one vine sample will have multiple species. These are what some of the plates look like once he has grown those out into larger spaces. But you can't just identify the species based on what the plates look like. So he then sends away each sample for DNA sequencing. And so what the results he gets back is a DNA sequence, and then he compares that sequence to an international database of known fungal species. This is on trunkdiseaseid.org. And so that's how he identifies which species belong to which samples. So this table is showing a snapshot of what genera we have found so far. So if you look over to the right at the legend, the arrow is pointing to pathogen for yellow. So I'm pointing out which genera are pathogenic, and this is again based on that database. And so if you look at the table, look for the yellow bars, Diaportha, Cytospora, and Phaeoechromonium are the 3 top pathogens that we have found so far. And you can see the numbers 40, 39, and 38 for the number of times we found those. You can see Botryopsera down on the list, which is a pathogen. We have only found that 3 times. So some of these results were somewhat surprising. I guess we thought we would find more Eutypa. Cytospora, that was for a plant pathologist, that was very exciting for them to find because we were not previously aware that that was present on grapevines in Minnesota, since this type of research had not been done before. So a summary of our findings so far and the implications of those. We know from this research that grapevine trunk diseases are very common in Minnesota and Wisconsin vineyards, and all 35 vineyards that we sampled did have a grapevine trunk disease pathogen in at least one sample. Some of the common species are listed here, similar to the table that I showed in the last graph. I wanted to point out something with Phaeoechromonium that's interesting and important, I think. It takes a longer time for this genus to be grown out in the lab on agar. And so there's a possibility that it might have been underdiagnosed previously, but Davey keeps the samples in the lab for weeks and weeks, even after he's sequenced the fungi off of them, to let them grow, because sometimes he lets them grow and all of a sudden we're seeing species that we didn't see before that are grapevine trunk disease species. So we might end up finding more Phaeoechromonium as we go on. It's also important to note that we've found grapevine trunk diseases in both unhealthy and healthy looking vines. So the implication of this may be that these vines can survive sometimes just fine, even if they have grapevine trunk diseases, depending on the overall health of the vines. Future projects, we are working on this right now, studying grapevine trunk diseases in nursery stock. So we're getting stock from 3 different nurseries, and we're going to be analyzing that in the lab, developing rapid diagnostic methods so he can get these results faster. And then the next big step of the project is researching and developing recommendations for managing grapevine trunk diseases in cold climate vineyards. So we want to find out what's the best time to prune during the winter in order to avoid grapevine trunk disease. Protecting pruning wounds. Is it really feasible for our growers to paint pruning wounds, or can we just recommend they paint larger wounds? And then test out this hot water treatment for nursery stock. There are a couple of papers that have found this effective, but it hasn't been studied on any of the cold climate hybrids. So we will be looking into that. All right, I'm gonna turn it over to Tim.
Okay, if anybody has any questions at this time, now's the time to put them in the question and answer section. And all right, so I will just get started here. And I'll share my screen.
Someone asked if we isolate only from discolored wood. No, we would isolate from any wood that we sample out in the vineyard.
Okay.
Because a few times we did find that even if it had no discoloration, there was still trunk disease present.
Okay, can everybody see my screen now? All right, so I'm Tim Martinson, and everywhere else, Richard Smart did come to our area, I believe it was last year or 2 years ago, and started talking about trunk disease. And my thought was, well, gee, this isn't anything new. We have vineyards that are 50 years old, 70 years old, 80 years old. And I think I'd like to find out, well, what are some of the impacts? So that's when we decided to do this survey. And I'd like to shout out to Janet Van Zoren, who did all the work and analyzed the results here. So I'm going to start out by showing you this picture of a vineyard that we ripped out last year. This is a Riesling vineyard that I did a lot of work on, and Alan Laxo before me. And it basically was 15 years old, and we were just done with it. And so when we cut off all those trunks, I took a look at and took pictures of several of them, trying to figure out, well, can I tell which ones have trunk injury and which ones don't have trunk injury? And so I'm looking at these, and I think I can identify with the blue circles maybe 3 or 4 trunks that I'm pretty sure are very healthy. I can identify a couple there with wedge-shaped cankers that are maybe indicative of Eutypa dieback, but the rest of them I'm just not absolutely sure about what's happening. There's a lot of discoloration, but I think any older trunk I've cut through in the Finger Lakes has some sort of discoloration. So I'm very confused about diagnosis. And I was talking to a grower who said, yeah, you know, I think 95% of my vines have trunk injury because when I cut them open, they're all discolored like this. So that leaves me a little bit confused about impact. So I'm very interested in measuring impact here. And so this is that vineyard before we cut it down, and it was a very productive vineyard up to its last year. Pruning weights between 3 and 5.5 pounds per vine. Those are big vines. And then the yield in our last cropping season, you know, somewhere between 10 and 15 pounds per vine. And that's grown on trunks that look like this. So they're apparently very healthy. We replaced the trunks many times as we pruned the vineyard, but it still turned out to be a pretty productive vineyard. So the question I've had, sort of before and since, are what is the impact in our vineyards in the Finger Lakes of trunk diseases, basically through their the effects that they have on the vines, which would include dead cord arms, blind buds on cane prune vines. Crown gall, another one that's not really a viral disease, but is very present. So that's the question we're addressing. How significant are trunk diseases in reducing canopy fill? And I have that asterisk there because we didn't really take a look at the pathogens to determine what the cause is. So there's lots of other causes that can lead to dead cordon arms, blind buds, and so forth, including winter injury. So what we decided to do in the spring of 2019 was do a survey of several blocks in 17 growers' vineyards. We looked at 32 native and hybrid blocks, natives being Concord, Catawba, Niagara, etc., Labrusca types, and then the other hybrids, which are mostly the older French hybrids. And then we also looked at 29 Vitis vinifera blocks. Varying ages, but the guess that I would hazard is that many of the native blocks and the hybrid blocks that we sampled have been in production somewhere between 20 and maybe even 50 years. So they're very old vineyards, and the vinifera ones are probably younger. So here's what we did. Here's our methodology. We took a look at 6 rows of 50 vines. That's 300 vines total, and we scored each vine in this 50 by 60 inch grid for 4 things. So we looked and tried to find presence of shoots that had foliar eutypa symptoms. We scored each vine for whether we could see any galls on them or other symptoms. And then we quantified the missing canopy. So we tried to look at each vine and figure out the percentage of the canopy was missing, either due to count nodes that didn't produce shoots, blank nodes on cane prune vinifera, or dead missing cordons. And then separately from this, we counted the number of missing vines. So that's the data I'm going to talk about. And I would just want to emphasize that we weren't after any particular trunk diseases or pathogens here. We did no pathogen identification. So this is just all about the spare parts and the missing canopy and its impact. So this is what I'll be reporting on. So here's how we scored the cordon or cane injury. We categorized it for each vine visually as 0, 25, 50, 75%, or 100% injury, leading to shoot reduction and cluster reduction, and the assumption that the amount missing would be proportional to yield in that vineyard. Okay, so I'm going to start with the type of dieback. And we looked at visual symptoms. We did not cut through any canes to look at the wood. But I'm going to show you a few box plots here. And what the box plots have is the box is 50% of the vineyards that we sampled, and the whiskers are encompass 90% of that population. So here I've split it out into vinifera, Concord, Niagara, and Catawba, and then other hybrids. And the bar through the middle of the middle box plot shows the median value. That means that 50% of the vineyards had more than that, and 50% had less. You can see it's 1.5%. The maximum value was 10%. And really, on the other hybrids and vinifera, the amount of visual eutypa symptoms was very, very minimal. So that was concentrated on Concord, Niagara, and Catawba, which tend to be our older blocks that have been in the ground for, say, 30 to 50 years or more. Crown gall was another story, and a much more serious issue in the Finger Lakes. And as you can see on the right here, it's much more severe on vinifera than on the natives and the hybrids, but it's still present in the other vineyards. So here's what we found in the vinifera. We found the median percentage of vines that have crown gall symptoms to be 5%. If you go up another 25% of the population that we surveyed, that's between 5 and 25% of the vines with crown gall. If you go up to 90%, that's 72% of the vines with crown gall. So there's 3 vineyards that you could see up there that had over 70% of the vines with crown gall, and that might be related to inferior sites or kind of marginal areas climate-wise that might be more susceptible to winter injury. So on the natives, the median was much lower, 0.3%, and we get up to the 75th quartile. That's 17%. So 75% of the vineyards had less than 17%, and then the top of that was 28%. On the other hybrids, much lower. Basically, the median was indistinguishable from 0 and the 75th quartile at 4%, and the 90th quantile at 21% of the vines with crown gall. So, more crown gall than Eutypa. Okay, so this shows all the vineyards we surveyed, and they're sorted into order of percentage of vines with various symptoms. And just want to go through that and show you again, this is what we saw in the previous slides. For natives and hybrids overall, median was 0.3%, the maximum 10% on that Concord vineyard, and the range from 0 to 10%. Median value of the blocks with visible galls was vines with visible galls was 0.3%, so half the vineyards had less than that. That's good news, and half had more than that. And you can see that's concentrated in a few varieties like Isabella, Niagara. There's a couple Niagara blocks, and Valvin Muscat, but not much in Concords or Elviras or Auroras. So then our estimates of missing canopy, again, median of 5%, high of 26% missing canopy. That would be sort of equivalent to 26% missing yield, and then 0.5% down at the bottom. Okay, for vinifera then, didn't see any foliar Eutypa. We did see a lot of crown galls, including one vineyard that had 94% of the vines with visible galls. Again, a median value of 5%. That means half the vineyards were pretty clean, and half, as you can see, had a fair amount more. And then when we go to the missing canopy, the median value 11%, and the range from 1 to 44% of missing canopy. That's a fairly wide range. So this takes into account all the vines that were in place, but it doesn't take into account the vines that were actually missing. So we have a separate tally for that. And here's the percentage of missing vines, the vines that had been ripped out. So again, in the natives, a median of 3.2 up to 16.7 was the range there. And that allowed us to add that onto the other missing canopy and come up with a revised estimate of 8.8% as the median value, and 35.8 as the maximum, and 1.3 as the minimum. So what does that translate into? If you're growing Cayuga White at $600 a ton and getting 6 tons per acre, fairly conservative yield for that variety, that would be costing you, if you're absolutely average, in the range of vineyards, $317 per acre per year. That's substantial. If you're growing Concord, even at $230 a ton with 8 tons per acre, that would amount to $160 per acre, which would be about what you might spend on your spray program. So it's a very significant loss. If we add the two together, the missing canopy from the vines that are still there, plus the missing vines, I can say from this survey that the missing vines added 3.8% crop loss. Again, 137 per acre for Cayuga, 70 per acre for Concord. For vinifera, much different story. When we add add them up, we come up with a median percent of missing canopy as 20.5%, adding on the missing vines. And the numbers here are not going to match up exactly, but if you're looking at Riesling at $1,600 a ton and 4 tons per acre, that's going to cost you almost a ton of grapes per year, 1305 per acre per year. So if we add those two together, then the missing vines calculated a slightly different way ended up costing $600 per acre, $608 per acre per year. So the impact of missing parts from our survey, which is, I admit, a very preliminary and simple-minded survey. Here we go. Eutypa, visible Eutypa at least, modest amounts, mostly on native grapes. Crown gall is the most significant pathogen that we've identified. With a smaller amount on the natives and hybrids, but a larger amount on the vinifera. That's undoubtedly affected by their relative cold hardiness and the winter injury that they get. Also, the grafting, because many of the natives and hybrids are ungrafted. So missing canopy, natives and hybrids, the halfway point, 5% range, up to 26%. Vinifera, median 11%. So missing vines, either 3.8 or 9.5%. So that's what we came up with our dead arms, our blind buds, and the missing vines. And I just want to summarize it here. This is feeling a little bit like overkill, but if we look at the different prices per grapes and the expected yields per acre on a good site, we're looking at those numbers. If you add on the percentage of missing canopy, then those are the losses that we came up with. If you talk about just the missing vines alone, then we come up with those numbers. And I want you to note that the missing vines is almost equivalent to the missing canopy in terms of an additive loss for people. So the total there that we've already done, and the percentage of this, okay, the natives and hybrids were together, so about 60-40 between the missing canopy and the missing vines, and the vinifera about 50-50 between missing vines and missing canopies. So a couple of things I want to close with here. First of all, these are median values, so half the vineyards would have had lower losses, but half had higher losses, and sometimes much, much higher. It would be 2, 3, 4 times greater for this, for certainly the vinifera people that had a lot of crown gall injury and a lot of missing canopy. And the second thing that I want to emphasize is that although this webinar is about trunk diseases, we didn't really actually go out and identify trunk diseases beyond the Eutypa and the crown gall that we were able to look at. So we don't know how much of this is attributable to fungal trunk diseases. I find it somewhat encouraging that we have Concord blocks that are 70, 80 years old, and they appear to still be very productive in spite of all the time that's allowed them to develop these things and allow the diseases to grow. But there's other things that certainly contributed to the missing canopy that we found. Finally, this just puts up the importance of regular vine renewal and replacement. If you have empty spots in your vineyards and you're still putting on the same inputs, you're definitely losing money. And when I look at the Concord losses on the natives of $162 per acre from missing vines and missing canopy, that is either equivalent to the fungicide program that they put on the 4 sprays, or it might be their actual profit margins. So while we pay a lot of attention to foliar diseases and put a lot of money into controlling those because they can be so potentially devastating, this is kind of a more quiet sort of loss that accumulates over years if you don't keep up with it. So I'm going to close here with saying that if you want a vineyard, your goal should be to have a full canopy and no missing vines, and that implies going through and replacing things at regular intervals. So with that, I'm going to turn this back over to Annie for her management section.
Okay. And Tim, feel free to jump in at any point during this management section as well. All right. There we go. All right, so again, I'm going to focus on management recommendations specifically for cold climates. There's a lot of information out there for management recommendations on trunk disease in warmer climate vineyards, and our climate, exemplified in this photo, can create some challenges for this. Specifically, one of the recommendations is to spray right after pruning in order to protect those pruning wounds. Well, we're pruning right now, so we can't do that, but also, the other question then is, do we really need to be spraying right after pruning if the diseases are dormant in these current temperatures? Those spores are not active until it gets up to about 35 degrees. So this leaves some really interesting management questions that still need to be answered. So I'm going to be talking about what we can recommend at this point, but just keep in mind that more research still needs to be done. The first sort of question that leads to a recommendation is, when would be the ideal time to prune during the dormant season in order to minimize the potential of trunk disease? The idea behind this is, once the pruning wounds dry up and heal, it's very difficult for any pathogenic fungi to get in there and infect that wood. So let's say maybe you're pruning in January and they're 6 weeks before it gets warm enough for those disease spores to be active in the air because you live in Minnesota or Wisconsin and there's snow on the ground and it's staying below freezing. Are those, for instance, those 6 weeks long enough to dry up those pruning wounds so that the spores cannot inoculate? There was research that's similar to this in British Columbia, and this slide is again from the Northern Grapes Project webinar in December of 2018. So if you were on that webinar, you've already seen this slide, but they were looking at pruning at different times throughout the growing season from December 15th monthly up to April 15th. And then they inoculated those pruning cuts on April 16th. And they wanted to see how easy or difficult it was for that grapevine trunk disease to infect those pruning wounds that were done at different points in the season. And indeed, they did find out that if they pruned earlier, January, they had 80% less infection than if they pruned later in April and then immediately inoculated those pruning wounds. So that's a question we want to look at in Minnesota, but what we can say right now for recommendations is you might consider pruning earlier if it is feasible, and if that makes sense for your area. Yeah. But we're still doing research on that.
Yeah, I want to jump in here, Annie, and talk about you know, the recommendations they have in California, because they've done a lot of work on grapevine trunk diseases, and there they recommend either late pruning or double pruning the vines, because the spores get released in the middle of the winter in the warmer, you know, central and coastal valleys there. And one of the things I got from Jose's talk was that in British Columbia, where we have colder winters, the spore release timing is just totally different than it is in California. So the practices they recommend for California don't really apply to, even in British Columbia on vinifera grapes. So that is a question I've had for a long time is, okay, if you prune in January, are those wounds going to heal up and prevent infection by the time the spores are released, presumably in April or May when it warms up?
Right. So I'm hoping that this is the next thing that we look at in our research. And Davey is interested in looking at this as well. And part of this question has to do with spore trapping. So you can put out this tool in the vineyard all throughout the growing season, or all throughout the dormant season or the growing season. And you can study when those disease spores are active. So it traps those spores and you can collect them every day or every week. And so we can see when are those spores becoming active in the spring, or when are they stopping activity in the fall. And that'll help us answer this question.
Annie, I got another question for you. Are you guys planning on inoculating, pruning at different times and then inoculating the trunks with spores?
I think that would be part of this. Yeah. Mm-hmm.
That sounds
Similar
like
to what they did in the British Columbia study. I think that's what he's going to try to replicate in our area. So we just need to find a vineyard that we can do this at that would be willing to collect those spores every day or every week. The second management practice that we hear about a lot is the use of protective sprays and latex paint. And so I've talked to growers from warmer areas, and they say that it's very common in the vineyards that they work at to treat all pruning cuts as they make them. One grower who moved to Minnesota from a California vineyard said that he had to paint every single pruning wound immediately after he pruned the vine. Of course, you might imagine that idea doesn't go over well in Minnesota. Growers who, some of them are growing as hobbies, some of them who are growing a lot more seriously, but regardless of who I'm talking to, they're not excited about going out there and treating all of their pruning cuts. But one thing that I do recommend that I do think is feasible is if you cut down a trunk or if you cut down a large cordon, you could go out and paint that cut. Let's say if it takes you 3 minutes to cut down a trunk, it might only take you 5 seconds to paint that pruning cut. So is it really adding that much time or effort? I think it would probably be worth it. But I still do want to do some research on this, because there's a question of, once the paint starts cracking, can the disease spores get in there and settle in? So this is something that you could try, but just keep in mind that we still need more data on this in our climate, I think. We're also interested in testing the efficacy of Raleigh or Topsin as a dormant spray in the spring after we prune, to protect those wounds. But it is difficult sometimes in our climate to get in the early spring just because of rain and snow melt. So that's definitely a challenge.
Agreed.
The other thing that we're going to be looking at is a hot water treatment for nursery stock. There were 3 previous studies that looked at this, and what it is basically putting the nursery stock in a hot water bath for a certain amount of time. It's something like 20 minutes, it's short. And the idea is that it will kill any disease fungal spores on that nursery stock without damaging the plants. And the studies where they did this had mixed results. One of them showed that while it was effective at killing the fungi on the nursery stock, that fungi is going to come right back once you put that stock out in the field again. So in that case, it didn't really seem to be worth it. But we do want to look at this in order to assist the nurseries and provide them more insight on whether or not this practice is going to be feasible or worth it for them. Current management tips that we have for cold climate vines in terms of trunk disease. The first one, and this is a big one, and it's something that a lot of our growers in my area are already doing because of winter injury. It's removing and replacing non-productive cordons. I'm going to go through all of these, but the second one is dabbing those large cuts with latex paint, like I talked about before. Third, remove cuttings from the field after pruning and burn those. And some growers do this and some don't, but I think this is a great practice, not just for trunk disease, but other diseases as well. 4, consider pruning earlier before the temperatures exceed 35 degrees. I know there are people on this webinar for which that recommendation might not be relevant because maybe you live in an area where it's regularly above freezing throughout the growing season. 5, keep vines healthy in order to slow the effects of grapevine trunk disease. If your vines are healthier, there's a chance that the trunk disease just isn't going to have as much of an impact. I did write an article on this last year where I described these in more detail, so the link for that is at the bottom. So that number 1 tip that I mentioned is retraining unproductive cordons and unproductive trunks. And this is something where some growers do have a hesitation about this, but if you look at this photo on the right, this is from a vineyard I visited in December. I see this a lot. They're keeping that cordon there, even though the cordon is only producing maybe 2 clusters right at the end of the cordon. That's not a cordon worth keeping. And so there's no shame in retraining that cordon. Get that off. Thanks for laughing, Tim. And there was data presented in the Northern Grapes Project webinar in 2018 that vines that were frequently retrained were found to have less grapevine trunk disease. So here we're cutting off that cordon. We're identifying a nice sucker with good bud placement coming out from the ground, and we are training that one down as a new cordon. Of course, when you do this, don't just put the whole cordon down at once. Only put down about 15 inches or so. Otherwise, the end of that new cordon is going to be weaker and less fruitful. Just develop it over a couple of years. Tip 5, keep your vines healthy. Grapevine trunk disease, if your vines are already stressed out, they're less able to fight diseases. It might just be able to take advantage more readily over those vines. If your vines are healthier, they're vigorous, they've got good fertility, they're happy, they've got a good sight, those disease effects may be less severe or more slow in affecting those vines. So things like fertility, keeping up with good pruning techniques and good crop load management, disease and insect pest control, and good site selection, all of these things are going to be really important. You know, as Tim showed in his data as well, just because it has trunk disease, that doesn't necessarily mean that it's going to be an unhealthy vine. So a couple major points on management. The first, do replace unproductive cordons and renew trunks and destroy that dead wood. Don't pull out vines if they're producing well. You know, in 2018, after Dr. Smart's visit, we did have a couple of vineyards start tearing out blocks of vines that were producing very well because they cut into them and saw trunk disease symptoms. I would say don't do that. If they're producing well, just let them grow and manage them. Consider pruning earlier rather than later for a lower chance of grapevine trunk disease infection, but we are going to be doing more research on this. Anything to add there, Tim?
Yes. Many of the pathogens that you're finding that are so common, you know, they're just ubiquitous. So the prospects of, you know, getting rid of them or worrying about them when your vineyard is still productive, I think that's the wrong tack to take. And I think, you know, to me, the most important thing is to go through regularly and systematically each year, find out the portions of your vine that, you know, are not being productive, or if you happen to have replacement wood that is just looks like it would be a good cordon replacement, I would replace those cordons sooner rather than later. And same way with trunks, I would try and leave a renewal spur down at the base of the trunk, let that grow up so that you always have a spare part. And it works well for us here in the Finger Lakes, and I think it will work well in cold climate areas too.
Absolutely. All right, so that's my last slide, and looks like we have time for questions.
Okay, so we had a couple of questions come in. There's one in the chat box that says, this is mainly for Annie. I've become aware of Pestilatiopsis as a fruit rot pathogen. I noticed that it was also one found in your chart. I knew this was possible. Do we know if there are any other trunk diseases that are also fruit rot diseases? And then furthermore, can it be controlled as a fruit rot disease if it is already present in the trunk?
So if it's present in the trunk, you're probably not going to be able to get rid of it unless you removed that vine and that was the only vine that was infected with it. But you can keep spraying for pesta, and the strobilurins are the group of fungicides that are typically recommended for that, as well as mancozeb. So I guess what I would recommend is if you're in the vineyard and you see symptoms that are consistent with pesta, Pestilentiopsis, sorry, I'm abbreviating it. If you see symptoms that are consistent with that, I would take a sample and submit it to one of the diagnostic labs. I wouldn't just take a sample of the fruit though, or just the cane. I would definitely include a sample of the cordon, if not the whole vine, with that and send that into the lab so that they can look at the wood and indicate on the submission form that you're interested in seeing if it's infected the wood as well. And then you can make management decisions based on that. But I'm guessing you're just going to have to keep managing that with your fungicide program. You're not going to get it out of the wood.
Yeah, Paul, your follow-up question, who is a diagnostic lab? We have one at Cornell. Of course, there's private diagnostic services as well. And the other part of your question, do you know if there are any other trunk diseases that always that are also fruit rot diseases? Yes, one of them is phomopsis. Phomopsis forms cankers on trunks and is always also a fruit rot disease.
Yeah. Oh, another thing that I wanted to mention, there are certain trunk diseases, and I can't remember which species they are, so I don't want to say something wrong, but if you see symptoms that look like streaking, that look like magnesium deficiency on the leaves, that can sometimes be a sign that you have trunk disease acting on the vines as well. So keep an eye on those symptoms, and just think critically about foliar symptoms. If you see something, before jumping to conclusions, think about all the possibilities that it could be, because trunk diseases are cutting off the vascular transport throughout the vines, and so sometimes they can affect the ability of the vines to take up nutrients. Just something else to think about.
Great. And then we also had a question come in on the question and answer thing that says, you kind of talked about this already a little bit, Annie, but does vine stress affect growth of the disease?
So I think in general, but I would want to talk to one of the plant pathologists to give more detail on that. Tim, do you have anything else to say on that?
You know, I'm not aware of any controlled studies about that,
Right.
but I would just presume that it's better to have a vine that's less stressed, and that would have some impact on these fungal pathogens that attack trunks.
If I remember correctly, in the December 2018 webinar, that was discussed, and I think the conclusion was there still needs to be more research to really quantify that. But I agree, just managing your vines well. If they're more vigorous, they're more healthy, they're putting out more good suckers that you can use to retrain, you're going to be better off.
Great.
Yeah, I saw another one on the chat bar. This is from Barry from UMass. Do you isolate only from discolored wood?
No, we isolate from non-discolored wood as well. Basically, any sample that we take in the vineyard, we're going to test that in the lab.
Great. We have another question. I've been painting with tree wound paint. Is latex paint better?
Not that I know of. Okay. The idea behind latex paint is you're just creating a physical barrier. That's all.
We have a bunch of questions coming in. Let's see.
It's just usually people have latex more readily available to them. Go ahead, Becky, sorry.
No, you're totally fine. I experienced a severe loss of cordon blind wood after the extreme 2012 drought. I replaced cordons to find a huge amount of Eutypa type wedge discolored cross section. I've since learned to discontinue bench cuts on old spurs. I have had much success with only pruning 1 or 2-year-old wood and leaving old spurs. It's not.
Okay.
So.
Thanks for the comment.
Yeah.
That's good.
How often is it okay to replace trunks? Does this stress the plant?
I know vineyards who replace the trunks every single year, as well as vineyards who do it every about 5 years or so. I think every single year is a little excessive. In that system, he has 4 trunks running up, and so he takes 1 trunk off every year, and each trunk survives 4 years. But generally, I think, you know, keeping spare parts like Tim was saying, but then if you're seeing that your cordons are starting to become unproductive, that's when I tell our growers that it's time to start replacing your trunk.
Yeah, I hope I pronounced your name right, Shruti. Yeah, my take on this is that if you lose the top of the vine but still have a healthy root system, that's sort of an imbalance, and so that can lead to a lot of large bull canes or several suckers coming out at the base of the vine. So so that's the only sort of stress I can think of that would be associated with replacing trunks, and that would be mainly if you sort of lost most of your vine but had a big root system, and so then you need to sort of have some control over how many shoots and shoots that you leave to retrain the vine.
Yeah.
Hope that answers you.
In Minnesota, when a lot of vineyards completely died back to the ground after our winter, we basically said leave at least 5 suckers coming out of the ground if the whole above ground part of the vine is dead, but if you have more than that, keep as many as you can.
Yeah, that sounds reasonable. And then pick the best ones for your replacement trunks. Do you want to do this other question, Becky?
Yeah, this other question is more of a comment. But I prune late in the season after the threat of a normal freeze, usually around March. As learned from conferences over the years, the fear of a late freeze loss is serious. So I think the question is balancing this risk of trunk disease infection with loss of buds from late season freeze.
Yeah.
Yep, absolutely.
Yeah.
It's a trade-off for sure.
Right. You can always cut off more later if too many push, but you can't paste them back on the vine. So
yeah.
if your varieties where you're growing are prone to winter injury, it's a good idea to check the bud mortality if you can before you prune, and adjust your pruning accordingly. And as Dave said, prune late in the season. Okay, I would just want to make a final comment. I think when this trunk injury business emerged with Dr. Smart's visit a couple of years ago, it got people sort of into a big panic. And I think the data that I have and that Annie has is sort of reassuring that, well, yes, it can be a serious problem, but no, it's not going to be something that really destroys a lot of vineyards. So I think the message I want to leave with people, and correct me if I'm wrong here, Annie, is that these are things that are just going to appear that we've dealt with for many, many years here in the Finger Lakes. I don't have any sense of whether they're getting worse or better, but they're something that you as vineyard managers are going to have to deal with and manage in your vineyards. But falls
go
part of an apocalypse, and it's not something that you shouldn't be concerned about either.
It's actually a good thing then that there's been more awareness drawn to this issue, because it has always been an issue. And it's kind of just as if now we're developing a better understanding of a problem that we've always had so that we can manage it better.
Yeah.
So, you know, that's kind of a positive spin on it. When I gave this or a similar talk at the Iowa Grape Growers Conference, somebody at the end said, well, what's the good news? And well, that's kind of the good news. You know, it's not a new problem. It's just a problem that we've drawn more attention to.
Right.
Okay, great. Annie, I'm going to stop your sharing so that I can pull up one last slide.
Great.
So thanks so much for joining us for today's webinar, and thank you, Annie, and thank you, Tim. That was really educational and really interesting. And you can check out our upcoming webinar, which will be in March, where Lynn Cisnowski is going to talk about growing grapes without glyphosate. And the registration link is listed there, and it also went out to the Northern Grapes listserv. So if you got the email about this webinar, you should have gotten the email about the other webinar, or this next webinar. Excuse me. Thank you so much.
Thanks, Becky.
Thanks for coming. It was fun talking.
All right. Well, I'm going to log out, and I'll talk to you guys later.
Great.
Hang out before you do, Becky. Is it possible to capture the registration list before you do, or not?
I don't know.
Okay.
But I'll figure it out.
Okay. Thanks.
Mm-hmm.
Annie, that was great. Thanks so much for doing this.